All right, so we're going to go over some of the material in the Biology 140 review handout. I'm not going to cover everything because Biology 140 is a prerequisite for this class. And I've also made my notes from Biology 140 available to you, so you can go ahead and access those and refresh yourself. But there's one important thing that is in there that becomes an important issue for what we'll be covering this semester, and I'm just going to go over really quick here. The types of cells in a mammal and us humans, we can classify them into two things initially, and that is we have the somatic cells and the sex cells, which we call the germ cells. So any cell type that is not involved in sexual reproduction is a somatic cell. Any cell type that's involved in sexual reproduction is the germ cells. Now, the number of chromosomes in somatic cells in humans is 46. And in germ cells, it could be 46 or 23. Now, in germ cells, when they through the process of meiosis, you go from 46 to 23. But okay, so what is 46? So 46, there are 22 autosomal chromosomes. So 22 autosomal chromosomes. That's also true for germ cells that have 46 chromosomes. Now, those 22, there's 22 that come from the 22 of the maternal version, and there's 22 of the paternal version. So you have 22 maternal and 22 paternal. And we'll say that and these are the autosomal chromosomes. Well, that leads us to 44. Where are the other two coming from? So the other two decide the biological sex of the organism. And this, for humans, it'll be the X and Y chromosome. So you receive your X chromosome from either your mom or your dad, and your Y chromosome can only come from your dad. So you have either X and Y, or X and X. And the X and the X would be the biological female, the X and the Y would be the biological male, and that will get us to 46. Now, when we're looking at somatic cells, every single one will have this whole set of chromosomes. So there's no missing chromosomes, there's no difference genetically between different cell types. So a neuron, which looks very much phenotypically different than, let's say, a liver cell or a muscle cell, will have the same set of chromosomes between them. And that is a very important point to keep track of later when we talk about differentiation a little bit and gene expression. And we'll cover that more in detail later. As far as the germ cells go, well, the ones that have the 22 paternal, 22 maternal, and the X and the X and the Y, or the X and the X, what will happen is they'll go through meiosis, and what you'll end up with is a combination of chromosomes 1 through 22 and either an X or a Y chromosome. And that will be the germ cell that is haploid. So in this case, all the somatic cells are definitely diploid, and all the germ cells, some of them will be diploid because they're the precursor cells that go through meiosis to create the gametes, or they're going to be haploid where they're the sperm and the egg, for example, in humans. And I think that's basically everything that I really would like you guys to know, and it's covered in the handout a bit. Another thing that we can talk about real quick is that the difference between genotype and phenotype. So, in the case of phenotype is basically a trait, the combination of traits that leads to something visible that can be measured. So it has to be a measurable thing. What do we mean by measurable? For example, height is a trait. Well, we can measure height. The amount of oxygen a person can take into their lungs, for example, that would be a measurable trait. You can measure that, etc. Blood pressure, things like that. But what about the cellular level? Well, the cellular level you can look at, well, it's something that you can measure the ability of a cell to metabolize glucose. and compare it to another cell type and say, well, the phenotype of this cell is that's very good at metabolizing glucose, whereas this other cell type is not that good at metabolizing glucose. So that's also a measurable trait because you can measure that in the lab. So if it's not measurable, you can't really call it a trait. I mean, you cannot. Now, what's the link between phenotype and genotype? Well, genotypes, so this is for phenotype. Phenotype it just comes from traits. Whereas genotype, well, we just looked at genotypes. Genotype comes from this, right? The genetic material in the cell. Now, the genotype of every cell in a particular individual is going to be similar. They're going to be the same because you'll have the same set of genes and etc. Between different individuals, for example, 2 different students, they might be different. They're going to be slightly different. And that goes to, for example, which allele each individual is carrying. So what are alleles? When we're looking at a gene, or when we're looking at a chromosome, let's imagine that this line right here is a single chromosome, right? And there's 2 versions of it. There's the maternal and the paternal, but we're just going to look at one. Let's say this is chromosome 1, paternal. So 1, paternal. On this chromosome, you'll have a bunch of genes. So here's like, for example, 1 gene. Here's maybe another gene. Here's another gene. Maybe another gene. So gene 1, gene 2, gene 3, gene 4, 5, 6. Of course, this is a very tiny 1 paternal. There's thousands of genes on each chromosome. But we're not going to draw 1,000 genes. That would be too much. So on the 1 maternal, you'll have the same exact thing. You'll have gene 1 here. In the same position on the chromosome, you'll have the same genes. The difference between the 1 paternal and the 1 maternal could be slight, meaning that this gene 1 might be slightly different than the gene 1 and the paternal. So the 1 maternal could be a little bit different. Let's say the 2 are the same. Let's say the 3 is slightly different. And let's say 6 is slightly different. And there could be many different versions of the same gene in the gene pool. So there could be another different version of, for example, gene 2 on chromosome 1 in the gene pool, meaning that there are other human beings that are carrying a different version of it. Now, what do we mean by different? And that's a big thing. What do we mean by different? Let's just look at on gene 1 in chromosome 1. So what we mean by different is that if I take gene 1 and I have it in an organism which has 2 copies of gene 1, okay, so it's homozygous for gene 1, that version of it, then I have a measurable trait that I see for this. So here is the trait. The trait could be anything. Let's say it's eye color or something like that. So I have the trait for this. But when I put the other version in a homozygous background, 1', I get a different trait. So we'll call this trait 1 here. I get trait 2. So let's say if this was eye color, trait 1 is blue eyes and trait 2 is brown eyes. So then I know that these 2 alleles, these 2 versions are different. Therefore, they're 2 different alleles. Just being slightly different in nucleotide code doesn't make you an allele. You actually have to have a difference in a trait when you put them in a homozygous background. So if, for example, I had 2 different versions of 1 and 1 double prime, when I put them in a homozygous background, I see no difference in the trait. Those 2 are the same allele. even though the nucleotide sequence could be slightly different. So these different alleles means that the genotype for this individual, for chromosome 1, the genotype would be 1 and 1 prime. Right? And you see that because the genes are sometimes named, they have names. So you'll see that, okay, this person is, the example that I have is sickle cell anemia in your handout. So HB beta, so you have HB beta S, and S being the sickle cell, and HB beta normal. So HBS for the sickle cell trait is a different allele than the wild type normal version of HB beta, which is part of the hemoglobin protein. And then you get into the whole thing of dominance and recessive genes. And that is something that is covered in the introductory material. It's not really important to what we're covering in this class. So I would say just kind of look it over and in the material just to make sure that you understand what that means. But just to maybe go over it, for example, with genotype 1 prime, let's say that under this heterozygous, when I have two different alleles, heterozygous genotype, I get trait 1. So if I get trait 1, I don't get an in-between trait or anything else. That means allele 1 is dominant over allele 1 prime because the trait 2 of 1 prime is not showing up. So in a heterozygous background, trait 1 is showing up, and that tells me allele 1 is dominant over allele 1 prime. Again, a trait has to be something that's measurable, otherwise it's useless. Okay, so I think this ends this video. If you guys have any questions, you know how to reach me, and I'll talk to you guys soon.